progress during the last 10 years that I'm going to cover. So the whole story will be about cells that come from the neurocrest lineage. And you know, neurocrest is a very important type of embryonic progenitors. They are often called the fourth germ layers because they do so many important things and contribute to so many tissues that people often say now that they should be considered like a new germ layer in addition to ectoderm, endoderm, and mesoderm. And this germ layer is unique to vertebrates. And these cells, they come from this neurocrest population. They associate themselves with the nerve. And because majority of them will give rise to Schwann cells, in the future, they're called Schwann cell precursors. And these Schwann cell precursors, they are really, in our opinion, neurocrest-like cells that are preserved on the innervation, and they serve like a stem cell pool. So I'm going to tell you a few things today about that. But before I will dive into these details that I think are very interesting in regard to storytelling, I wanted to sort of set up some broader scope, which might be good for students, which might be good for the physicists who joined our webinar today. And I'm going to say that just historically speaking, we always consider the nervous system, and specifically the peripheral nervous system, as something that forms the information exchange, right, in our body. It runs the action potentials, basically the electricity back and forth, transmitting sensory and motor information. And because of that, we can kind of control our body. We can sense things. We can actuate our movements, and so on and so forth. And this is the major function of peripheral nervous system. But during the recent years, there is a paradigm shift. We start to see that there are lots of very interesting non-canonical functions of the nerves that go everywhere in our body. And these non-canonical functions, they include, for example, the modulation of the immune system. They include, for example, providing morphogenetic information on cellular sources during embryonic development stage. And also the nerves are very well known now and recognized for controlling and enabling the regeneration of limbs, inner organs in different model systems, and hopefully partly in humans as well. So it's a quite hot topic, actually, the role of the innervation hosting these peripheral glial cells that resemble in their kind of capacity to some extent the multipotent neurocrest in so many processes beyond the transmission of action potentials and exchanging sensory and motor information. And this is a very convenient system for any sort of regulation like that, because these nerves, as you can see here on the slide, they travel everywhere in the body. They're found almost everywhere. It's hard to find a location which is not innervated, in fact. Probably the brain. Even the brain is innervated, actually, by peripheral nerve fibers, at least some parts of the brain, which is also quite interesting. And you can see here, these are three-dimensional reconstructions of mouse embryo at embryonic day 10.5. And already at day 10.5, you can see all these nerves extending to the limbs and inner organs, right? So already very early during the development, the nerves are reaching out to their targets. And they can change a lot up to the course of development targets. So this is sort of like a small introduction on that. And yes, as I mentioned, also the nerves and the glial cells around them, they play a very important role in regeneration. Actually, this is a very well-known model of salamander limb regeneration. It's a vertebrate model. If the limb is amputated, it comes back actually in 6 to 9 weeks. Can you imagine the capacity to regrow the limb entirely within several weeks? It will redevelop to the very high precision and accuracy. And in 2007, there was an absolutely seminal paper from Anup Kumar and Jeremy Brock's lab, where they demonstrated that actually the factor that triggers the regeneration is in fact rooted in the innervation, is in fact in these glial cells that are nerve associated, because these glial cells, they will generate the protein that is called NAG, and this protein will trigger the regenerative blastema formation and the whole reinitiation of the whole morphogenesis. So if actually within the amputated limb, the nerve is removed, if there is a denervation, then there will be no regeneration unless this NAG DNA will be sort of installed in the tissue that will form the blastema. So, and is absolutely essential. And these axons must be covered by the Schwann cells that secrete NAG. So you see, the glial cells, they play also a major role in inducing the regeneration in vertebrates. Besides this, the Schwann cells and the glial cells along the nerves, they're very important in development and dissemination of several important diseases. And one key disease is, in fact, is leprosy. So what happens in leprosy, actually, the mycobacteria, they infect primarily Schwann cells. So you can see here mycobacteria like invading Schwann cells, wrapping axons. And then what is happening, they reprogram them. They reprogram Schwann cells to stem-like cells. So this actually shows that Schwann cells, they're very susceptible to triggering some sort of stem cell fate, something like close to the neurocrest. And then these cells become migratory. They travel all along the body. They leave the axons, being infected by this leprobacteria, mycobacteria, and they disseminate the infection. There was actually a bunch of big papers in recent years addressing actually how mycobacteria causing leprosy, they impinge on Schwann cells, causing the leprosy disease. This is absolutely something amazing. Now we would like to learn how they do it on the molecular level, how they do this sort of like, how they induce this plasticity in Schwann cells. So now they're called kind of mighty bugs, and there is a big focus on them, looking into these transformations. Besides this, Schwann cells, peripheral glial cells can cause transmittable cancer. And transmissible cancers is something very rare, but they can be extraordinarily horrible when we find them in the wild nature. And probably all of you are aware of the case that led almost to the complete extinction of a species on our planet. These are Tasmanian devils living in Australia. So Tasmanian devils, they acquired the transmissible cancer of Schwann cell origin. And because these animals, they have very aggressive behavior, and they always bite each other. Like once they bite, these malignant Schwann cells, they come with the saliva, and they induce cancer in the new host. So the whole population of Tasmanian devils was rescued basically because several healthy animals, they were basically confined on an island where they could kind of live as an isolated population, and lots of other animals in the wild died meanwhile from this transmissible cancer. So again, the Schwann cells, they became converted into something more like cancer and stem. So like lots of interesting things to learn from that, I think. So when it comes to the development of these peripheral glial cells, they come in several flavors also as an adult population. They come as myelinating fibers that will insulate the nerves and will serve for the fast propagation of action potential, right? They will kind of wrap up the fibers, generate the myelin sheaths, or they will form remark bundles that are non-myelinating, but they're still very important to nurture the neurons and axons. And the history of these adult fully differentiated cells includes sort of like the previous stage, which is called mature Schwann cell, and then there are dividing progenitors, fastly dividing progenitors that are called Schwann cell precursors, and before them will be neurocrest cells. So the neurocrest cells will migrate and associate with the nerve fibers, forming the Schwann cell precursors. And we think that these Schwann cell precursors are really interesting cells. This is sort of like a subtype that is multipotent and contributes to development and potentially to the regeneration in many important ways. So the rest of my lecture will largely focus on these Schwann cell progenitors and where do they come from and what they can do, okay? So coming back to the origin, right? Neurocrest cells. Neurocrest cells form in the dorsal neural tube, and they will migrate from the dorsal neural tube to different destinations in the embryo. And they will give rise to mesenchymal fates, mostly in the head. You can see here the red head of an embryo, right? Heavily populated by the red neurocrest cells. Our face is fully made by the neurocrest cells, basically cartilage, bone, dermis of our face. This is made by the cells that exit the neural tube. This is absolutely surprising that cells exiting the sort of primordium of our brain in the end will make the skeletal cells, and they will kind of shape the jaws and the facial appendages. That's just incredible. But in the trunk part, they will not give rise to these mesenchymal fates. They will give rise to other derivatives which are mostly belonging to the peripheral nervous system. So those include sensory neurons in the dorsal root ganglia. These neurons, they reside next to the spinal cord. They will be like forming here and here on the sides. And these neurons, they allow you to feel the touch, the pain, the temperature, for example, vibration. So all these fine sensations. So we feel them, and we can position our body. We can feel the position of our body parts in space because of these neurons, because they also are responsible for the proprioception, because they would innervate the ligaments, they will innervate tendons, and they will innervate parts of the muscles that are responsible for the proprioceptions. And besides that, of course, the neurocrest cells will contribute eventually to the autonomic nervous system that is represented by the sympathetic and parasympathetic neurons that control the operation of our inner organs. And this will also include enteric nervous system of the gut, the motility of the gut, control of the motility of the gut, and this will also include neuroendocrine cells, for example, chromatin cells in the adrenal glands that release a lot of adrenaline and noradrenaline into the blood flow when we are stressed, and we need to have this, very fast, fight and run response to something. When you feel adrenaline like kind of boiling in your blood, these are chromatin cells in action, and they're also derived from the neuron. But as we know now, this is not exactly how the story goes. And that was exactly the mystery that we were solving during the last 10 years of work in my lab and in the labs of my colleagues. And now we will be slowly shifting to those stories. But before we will shift to that, I can tell you something more about the formation of these Schwann cell precursors and the type of the analysis that we apply to study them. So, and now we use actually a lot of single cell transcriptomics. Single cell transcriptomics is a very powerful technology. This technology is based on our capacity to isolate cells, like any cells from any organism, from any location, and take all RNA content from this cell and sequence it very deeply, individually, cell by cell. And in these massive experiments, we can sequence hundreds and thousands and tens of thousands of the cells like that individually. And then we can compare them. You can see here, every dot here, every colored circle represents the transcriptome of the individual cell. And then we can compare them and cluster them according to specific algorithms. So, and those cells that cluster together, that basically form what we call a cluster, these are the cells in a certain kind of specific transcriptional state, probably a cell type or a subtype of embryonic progenitor. And if we observe the process of development, then we will see how these cells change, how they gradually transform in something kind of more novel and fate-restricted, for example. And this is exactly what we see here. We see how the pre-migratory neurocrest transforms into the lamination phase when the neurocrest exits the neural tube, how the cells migrate to generate all these different cell types, and the set of fate split points where they decide to become autonomic nervous system or mesenchyme or sensory neurons, or they will become Schwann cell precursors, they will become glial progenitors. And if you look here, actually, if you look at this population of these kind of light blue cells, you will see that they occupy the central part of this plot. So this is sort of the kind of the average of everything. This is sort of like the prototypical signature of the neurocrest. Because if you ask me what is neurocrest, what defines neurocrest, it's very hard to give a very straightforward definition of the neurocrest having this data, because neurocrest cells are transient. This is a transient population, and you can see here that this transient population is changing continuously. So these cells, they're always on the go, they're always changing, transforming, picking the fate, choosing, differentiating, and so on, switching on and off different modules. Of course, there is something in common in all these cells, but at the same time, there are lots of differences. And the only population that sort of conserves the most conserved features and does not change much when the time goes by are those Schwann cell precursors, the progenitors of the future adult glial cells that will associate themselves with the innervation, will reside in the nerves, and they will most likely retain the multipotency, the potential of this mother neurocrest population. So now we are coming up to a concept, to a new concept that we are trying to establish. And I would like, probably this is the most important slide during my talk, simply because this is the most conceptual slide. Everything else is sort of like technical details and some particular things that might be neat and interesting, but this is the conclusion that I would like to serve in the beginning of my lecture, because it's just easier to remember. If we think about the process of embryonic development or regeneration or something like that, then of course, if we have a set of the homogeneous progenitors in some specific location, how could we create the cell type heterogeneity? How can we create different cell types here? We need to make a symmetry break. We need to some sort of a crosstalk in these cells or some autonomic memory in these cells. So these progenitors will generate the diversity of cell types in some location, right? They are programmed to do this. They have a capacity for this symmetry break, whatever causes this symmetry break. But sometimes in many locations in the body, local progenitors cannot make the symmetry breaks good enough to generate specific cell types that are needed there. And in that case, the nature must rely on completely different principle for making these cells which are needed to happen in that place. And often this is solved via the cell migration, for example. We know so many cases when cells migrate from the places quite distant from the destination, right? We know the example of tangentially migrating interneurons that cross large distances during the development of our brain. We know that cells building the vasculature, they can migrate quite a lot. All of you know different examples. This is quite a common principle. It's sort of a trick to complement the local tissue with the cells that are needed there, cell migration. But cell migration has many limitations. For efficient cell migration, cells must know where to go. So you need to sort of wire in, you need to embed some principles guiding, directing the cell migration. You need to set up the set of landmarks in the terrain where cells will migrate. You need to install the navigation machinery in these cells, and this is not simple. It needs to take some time to evolve and so on. It's extraordinarily complex and intellectual, and cell migration doesn't work on very large distance because the noise is too high, cells need to migrate for too long, lots of things can perturb this process. So typically migration happens while the embryos or regenerating tissue is tiny. So, but how this can be solved if the tissue is large, for example? Then the answer comes from our concept. There is infrastructure, right? Like vessels and nerves that penetrate everything. And they have, for example, nerve associated cells, these kind of brownish cells that cling to the nerve, and they may use the nerve as a navigation line. The nerves know how to navigate because our tissues, they must be innervated correctly. Evolution took a good care of the innervation navigation, because if our fingers or any other body parts or inner organs cannot be innervated properly, they are dysfunctional. So navigation in neurons is a very old thing. And these cells, they basically just use it. So they don't need to have probably their own navigation principle. They just follow the nerve, they overpopulate it. And then what can happen, somewhere in the embryo, the signal might come that will recruit these cells, and they will separate from the nerve fiber, for instance, and they will generate the cell type which is needed in that location. And we call this concept targeted recruited of Schwann cell precursors into the what is possible by the neurocrest phase. So, and we published a bunch of papers. We made quite a nice set of discoveries in, like, starting from 2009 paper where Jorge also participated. So we tried to expand this concept as much as we could, and now I'm going to tell you the story how we expanded this concept in the next 20 minutes of the time that I have. Everything started with a very peculiar experiment. It came like a pure luck, actually, the initial discovery. So we were studying the development of the peripheral nervous system in chick embryos by changing the transcriptional code that was supposed to change the fate of the sensory neurons in this dorsal root ganglia. So we were electroporating chickens with different constructs. And eventually we spotted that some of the electroporated neurocrest cells, they would migrate along the nerve, and they would separate at the nerve, and they would start to express transcriptional factors that are not typical for the Schwann cells or for the sensory neurons. They would express the transcriptional factors that are the property of melanocytes, pigment cells, like meat, for example. And then we could trace these cells going along the nerves, and we could see how they generate pigmentation. So it was an extraordinary, surprising finding that actually we were studying something in the nervous system, and then just because we were introducing fluorescent proteins in the cells, we could actually see with our own eyes how in these embryos cells migrate, how they separate from the nerves, and they become something completely different. So that was sort of a big change in the field of how pigment cells get born, because before the old concept was that neurocrest cells would just form and they would just immediately migrate to the skin and they will generate all pigment cells in the skin. But here it turned out that the major source of pigment cells are the nerves, and you can see it in this schematic here under the letter N, so that the primary cells like can seed some minor amounts of melanocytes by free migration, but then the nerve will develop, and cells covering the nerve, they will separate from the nerve and they will convert into melanocytes. And this will be the major source of melanocytes, and all nerves coming to the skin, they will generate melanocytes. So it was absolutely mind-boggling discovery that we managed actually to validate in mouse with the genetic lineage tracing and many other experiments. Now it's a big field. There are many papers confirming that this is happening in all vertebrate model systems, including lamprey, cyclostomes, right? A very basal chordate model. It is also happening in zebrafish, and so on and so forth. And what was probably quite interesting is that there are sometimes mutations in animals and in birds when the meat of an animal and all internal organs, they start to be black. They're just completely black. The animal from inside looks black. And one breed of such chicken is called silky fowl chicken. So this is a very popular chicken in Asia because the meat of this chicken looks black, very unusual. And it's just a bit extravagant. And what is happening in these chickens, they have a mutation in the gene of endothelin-3, and endothelin-3 is expressed not only in skin, recruiting these Schwann cell precursors into melanocyte phase, but it is also expressed in all inner organs and muscles. The expression becomes general, and it leads to the massive recruitment of Schwann cell precursors from the inner body nerves, and they convert into melanocytes, and those proliferate, and the whole all inner organs and meat of these animals start to be black. And melanocytes, they're very inert cells, so they don't damage any physiology. They just stay there, and they just stain the tissue black. So yes, it's a very peculiar thing. And another important kind of meaning for the human health is related to the fact that melanocytes in the human body, they are found in the inner ear, and they're found also in the heart. And it was not clear how melanocytes get into the inner ear or in the heart, because these are locations which are very far away from skin. And now we know, we just sent out a paper to a journal where we show actually that there are very specific nerves that come to innervate the inner ear, and there are specific autonomic nerves that come to innervate the heart, and they bring Schwann cell precursors that get recruited in these locations, and they generate melanocytes in the inner ear and in the heart. And if you ask me, why do we even need melanocytes in the inner ear or heart, I can tell you that actually they have extraordinary physiological importance, because melanocytes in the heart, they condition the media, the liquid in the inner ear. And if humans or animals, because of some mutation, do not have melanocytes there, it causes complete deafness. So these animals or people cannot hear anything. And when it comes to the heart, if melanocytes do not arrive to the heart, and the cardiac valves are not supplemented with melanocytes, they are too weak, and different cardiac insufficiencies develop with time. This is also something very unexpected and very interesting that came up in recent years. So melanocytes and the way how they get recruited from Schwann cell precursors along the innervation is completely new way of looking at the development of the pigmentation in the body. Then after we had this story, we thought that, well, what if these cells along the nerves, these Schwann cell precursors, they are so multipotent that they generate other cell types, not only melanocytes. There are probably some other like locations to which they complement. And we started to think actually, well, we know that there are two compartments in the autonomic nervous system. One is called sympathetic nervous system, is this one on the left, and there is a parasympathetic, green one on the right. And the sympathetic nervous system is very well studied. We know that these ganglia of the sympathetic chain, they have their own specific anatomical location quite close to the spinal cord. And they form from the migrating neurocrest cells quite fast after the cells start to migrate. They just come out of the neural tube and they go down to the dorsal aorta and start to make this ganglia of the sympathetic chain, pam, 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 very fast. This is the earliest derivative of the migrating neurocrest. And this happens at around embryonic day 10. The embryo is small and migration is easy. And then after these neurons, sympathetic neurons form, they have enough time to grow their processes, to target slowly developing organs. They take their time to mature and to find their targets. The situation is completely different when it comes to the parasympathetic nervous system. In parasympathetic nervous system, everything is exactly the opposite to the sympathetic nervous system. First neurons of parasympathetic nervous system are found after embryonic day 12.5, and typically they will be sitting next to the developing organ, or they will be within the developing organ. And it means that they appear so late, and presumably they should come from the neurocrest cells, but there are no neurocrest cells at these late time points when these organs are developing so strongly. Neurocrest cells typically migrate at embryonic day 9.5, latest 10, but there is a huge time difference, 48 hours or even more between the migration stages and when the first parasympathetic neurons form. And we generated this idea. We were just sitting and brainstorming in the lab. What if it works like with melanocytes? What if it works like that? The neurons inside of the brainstem and the sacral region of the spinal cord, they project towards the future ganglia of parasympathetic, where the parasympathetic neurons will reside. And then these nerves, they become covered by the Schwann cell precursors. So, but when the Schwann cell precursors arrive closer to the developing organ or tissue, they can be just recruited, and they will generate a parasympathetic neuron that will be immediately innervated by this incoming fiber. And that was a very kind of bold hypothesis, and we spent a long time addressing it actually. So for this, we used like lots of things. We used different clearing techniques, looking where the preganglionic and other different nerves go. We were really trying to understand the anatomy of the developing preganglionic visceral motor fibers quite well using these methods. And then we were also using quite a lot of genetic lineage tracing and whole mount characterizations. So since we could make these embryos very transparent, we could make sure that at very early time points after neurocrest cells just finish their migration, they will be sitting on the surface of the nerves. There will be no freely migrating cells. All these blue cells, you can see they cover the nerve. There is nothing in blue that migrates at this time point. And if we cause the genetic recombination, right? If we label the cell with genetic recombination permanently, and all the progeny of the cell will be labeled, right? Using this Glia system, genetic system for the lineage tracing. Then what we can achieve, if we label the cells along the nerve, we can track what happens to them in the future. And if we activate the tracking after neurocrest cells is not migrating anymore and sitting on the nerves, and we are tracking Schwann cell precursors, we could see later on at the late developmental stages whether these cells contribute to the parasympathetic neurons within the ganglia. So we had a look at all parasympathetic ganglia happening in the body. And what we found something very astonishing. We found that all these neurons are green. They express fluorescent proteins. So all blue cells are actually green, for the very rare exceptions. And these are the parasympathetic neurons. So which means that parasympathetic neurons develop from the cells that are nerve associated already after neurocrest cells finish their migration and finish being the neurocrest cells. If we do the activation of this tracking very late, like embryonic day 15.5, then we have exactly the opposite picture. All these neurons, they stay blue or purple, but they're not green. So they're not lineage traced for the very few exceptions, which means that from that late stage, the neurons cannot be generated from Schwann cell precursors anymore. So that was the most key experiment that showed that actually nerve-associated cells, they generate the parasympathetic neurons, and our idea was correct. Another experiment that we did was different. So we decided that if we take these nerves that come and bring the Schwann cell precursors to the location where the future ganglia will develop, what if we redirect or ablate these nerves with some genetic mutation? What will happen to the ganglia? Will they disappear because Schwann cell precursors will never be delivered by this nerve or not? So there was another interesting assumption. So we just wanted to sort of rewire the nerves So they go to different places. And then we could see if the ganglion that will be innervated by this nerve will depend on the presence of the nerve and the correct navigation and the presence of Schwann-cell precursors. And indeed, that was exactly the case. On the left panels, you see the controls. And the ganglia forming at the tips of these nerves, they are red. You can see these nerve-associated beautiful red cells. And if we use the mutation in red gene, if we substitute red receptor with 2 CFP proteins, like in both locuses, and there is no more red, then what is happening that some of these preganglionic nerve fibers, they disappear. You can see that greater superficial petrosal nerve that you can see here is not present here. It completely disappears. And these red cells forming the coalescing ganglion, they are also disappearing. So all the nerves like Jacobson nerve and so on, they are disappearing, and the ganglia are also disappearing. And this is happening because we target the nerve, because the nerve is gone or the nerve is misnavigated. Of course, we did many more experiments in that story. So that was quite a good story that we published in Science in 2014. But that was some sort of a conclusion image that some preganglionic motor nerves, they will bring Schwann-cell precursors on their surface. And then these Schwann-cell precursors will be recruited into the fate of postganglionic parasympathetic neurons. And these neurons will be immediately innervated by the same nerve that delivered the Schwann-cell precursors. So that was some new concept in neurobiology, because it looks like it's a self-propagating developing network of neurons where one neuron brings the progenitors for the next neuron in line, for the postsynaptic neuron. So it was kind of the chain of like sort of developmental connections and synaptic contacts. So it was quite peculiar to see that. Then we thought that, okay, let's move on. That was another cool story. How can we expand that? And we started to think about the development of adrenal gland, because adrenal gland had quite a known origin. Adrenal medulla had quite a known origin at the time. Everybody believed that cells of adrenal medulla, chromatin cells that produce adrenaline and noradrenaline, sitting here inside of the adrenal gland, they are also developing directly from migrating neurocrest cells. But these cells, like also, for the first time, they appear very late in an embryo. The embryo is huge, and neurocrest cells, they cease their existence for like more than 2 days already. So the concept was very similar to parasympathetic neurons. We said, like, what if chromatin cells are derived actually in exactly the same fashion? What if the visceral motor nerve brings actually Schwann-cell precursors towards the developing adrenal cortex, and then the cells are recruited and converted into these chromatin cells? So we did a bunch of experiments also, but here we started with single cell transcriptomics. So we were dissecting the areas from developing embryos to track actually what's going on in different stages. We made enzymatic dissociations, we FACS sorted them or did a 10x, and we did sequencing and clustering of the cell types to see which cell identities and cell transitions we observed during the development of the neurocrest, Schwann-cell precursors, and adrenal cells. And that's what we found. Actually, that was quite a cool thing to see. We found that actually there are 3 major clusters at 2 stages: embryonic day 12.5, embryonic day 13.5. So you can see that the blue and green cluster, these are Schwann-cell precursors, dividing and non-dividing. The green ones, they're outclustered a bit because they are dividing, and the signature, the gene expression related to proliferation, just moves them a bit down from the non-dividing Schwann-cell precursors. And then you see an interesting structure that is marked in red. And this red structure, these are 2 independent experiments, 2 different stages. It looks like a breach. So it's like a breach leading to the population of green cells. And the green cells, they express all markers of chromatin cells. They express TH, they express chromogranin B, and so on and so forth. So it seems that the Schwann-cell precursors within the developing adrenal medulla, they are actually connected with chromatin cells via this breach. And now if you look at sympathoblasts, the sympathoblasts are not robustly connected with anything, because most of them will be derived from the neurocrest cells directly. So immediately we had this hypothesis that Schwann-cell precursors, via this breach stage, they generate chromatin cells. So they are recruited from the nerves and they convert into chromatin cells. And we did quite a substantial analysis of all transcriptional factors, signaling molecules operating on that way. And we found out that this breach stage is not just transition from A to B, but these breach cells, they have their own transcriptional program. They express the receptors to serotonin, they express delta 3, they express THBDs, they express some genes that enriched only in these cells. So this is a very transient progenitor type, but this is an independent progenitor type. This is not just the blend of gene expression program of Schwann cell precursors and chromatin cells. It's not that just this gets down-regulated and this gets up-regulated, and that's why we get this breach forming. This breach is something on its own, some programmed transition that has its own genes characterizing it. That was so cool. And then we started to search for these breach cells. So for this, we use this HTR3A EGFP mice. You can see here that HTR3A is a receptor to serotonin expressed in this breach population of cells here. So we started to localize the cells in the tissue. And we could see that during the early stages, these breach cells, they appear like nerve associated, separating from the nerve, and they can blend with SOX10, which is the marker of Schwann cell precursors. And many cells are dual positive because Schwann cell precursors will switch on the breach program. The GFP has a long lifetime, so they will stay green. And then the GFP will be degrading when they convert into chromatin TH positive cells. So we could really look where these breach cells form and how they are seeded. And we could kind of really deeply analyze the transitions in the breach. So these breach cells, they really do exist. This is not our imagination. And these breach cells, they show the transient gene expression profile shared between Schwann cell precursors and chromatin cells, plus breach cell-specific genetic program. So, of course, then we used another approaches like classical lineage tracing. And what we did here, we just used PLP1, CRT2. PLP1 is the marker for Schwann cell precursors along the nerves. And if we label them with mutation activating expression of fluorescent protein permanently, so the progeny inherits this mutation, we will see which cells are derived from the Schwann cell precursors. And you can see this is a red adrenal medulla. It sits inside of this kind of greenish dim cortex. And inside of this adrenal medulla, there are lots of cells that are red and green at the same time, so they appear yellow. And in the sympathetic ganglia that is associated with adrenal gland, this is not the case. You see that the green largely non-overlaps with the red. And it was a huge amount of the chromatin cells that appear traced, which means they are derived from Schwann cell precursors. And if we kill the cells with diphtheria toxin activated genetically in the nerve before adrenal medulla starts to form, that's what we see. This is the control medulla, and that's what is left of the medulla when we do this killing. Of course, we cannot kill all 100% of cells because it's not entirely efficient process, but we can kill enough to see that there is a strong effect on the size of adrenal medulla, which means if Schwann cell precursors give rise to chromatin cells, if this is true, if we kill Schwann cell precursors, then chromatin cells must decrease their numbers. And that's exactly what this experiment is showing. And finally, we did a similar experiment to what we did with parasympathetic nervous system. We decided that we will ablate this nerve. You see this red nerve in F. We will kill the preganglionic neurons, and we utilized HB9-3-IL2-DTA genetic construct, very complex crossing of mice. So these neurons die, or they never form initially. And then this nerve is not forming. And when this nerve is not forming, then adrenal medulla is super tiny or non-existent. So you can see it here. This is the control adrenal medulla in C, and in D, you can see what is left of adrenal medulla if the majority of this neurons is killed. Again, we cannot kill them 100%. We can kill them maybe 90% only. We see that in B, there are some leftovers, very few neurons stay, and they will probably generate these very few leftover cells. But the phenotype is massive. The reduction is very strong, which means that indeed, again, this is additional confirmation that Schwann cell precursors are recruited from these preganglionic nerves and generate very important cell types for our body, controlling the operation of our inner organs and controlling our stress responses and normal well-being. So basically, this is the schematics that outlines how this process is happening, that primarily neurocrest cells come out of the dorsal neural tube, they will migrate, and they will seed the sympathetic ganglia. At the same time, these preganglionic neurons in the neural tube, they will start growing axons and they will start kind of extending to the periphery. And a lot of neurocrest cells from the late waves, they will associate with them because they will need to myelinate them in the future. But while they stay in the multipotent state on the nerve, they will be also recruited into the adrenal medulla. and they will generate majority of chromatin cells inside of the adrenal. And then later on, most of the cells will still differentiate into mature Schwann cells. You can see that they stay multipotent, they will contribute to the other cell types and tissues and organs, but then majority of them will generate normal Schwann cells. This is a very cool concept. And we can have a look at it clonally. We can see the clusters, how the clusters of colored cells are born. And by using this analysis, assessing the size of these clonal clusters, we can say that you need a lot of Schwann cells, individual Schwann cells, to be recruited, to be converted into these neurons. There is no situation like in a stem cell niche when you need maybe just very few stem cells, but then you can expand them through the amplification cascades, and then they will generate lots of progeny. Very few cells can generate lots of progeny. Here, this is not the case. This clonal analysis tells us that to generate 4 to 6 chromatin cells, you need to recruit 1 or 2 Schwann cell precursors. So it's a ratio of like 1 to 2, 1 to 4 maximum. So it's in the abundance of the innervation during the development is really important for this process. So this is the concluding slide for the adrenal medulla case. So we have the nerves, which are kind of depicted here as roads. And there are cars going along these roads. These are Schwann cell precursors that inhabit these nerves and travel along them and multiply along them. And sometimes these cars, they take a route of the innervation. It goes to innervate, like, for example, adrenal medulla. And then some cars will end up as chromatin cells according to this diagram. Okay. So this is almost the last concluding slide in my lecture. While the embryo is super tiny and small, neurocrest cells born in the dorsal neural tube, they can do free migration and they will create some structures, including some sympathetic neurons and so on, what is needed at this stage. So this will be done according to the free migration. Some melanocytes, some sensory, some autonomic neurons will be born, and lots of mesenchymal derivatives like a bone and cartilage of the face. But later on, the cells, lots of them, they will settle on the innervation. They will become Schwann cell precursors, and they will keep spreading in the body via the nerves, and the body is large already at that time point. And then it will be possible to recruit these cells from the nerve into other fates. And then you can get even more melanocytes, you can get enteric, parasympathetic, neuroendocrine chromatin cells, and many more cell types that we keep discovering arising from this very interesting cell source. Of course, the key task right now, setting up next big question is the regulation of the system, which signals define the fate of a Schwann cell precursor when it gets recruited. Can we take basically a portion of the innervation from the human skin and convert these Schwann cell precursors into cell types that we want to obtain, for example, for regenerative medicine or for just fundamental science? This is a very interesting task. And we keep working in that direction, but so far we have only very little progress because understanding molecular mechanisms is very complicated, extraordinarily hard, tedious, time-taking, and probably next 10 years we will make some progress along these lines. And with these words, I would like to thank my group, both in Vienna and in Stockholm, our key collaborators and funding agencies, and thanks a lot for your attention. I'm a little bit too late, like 5 minutes, but I hope we still have something like 15 minutes for productive discussion. Thank you. Please start asking your questions.
Okay, if somebody wants to ask any question, just unmute the microphone and ask, so we can stop with this presentation.
Hi, Igor. This is Lorena Rela from the School of Medicine at the University of Buenos Aires. It was an amazing talk. Thank you very much. I have a question regarding what happens to the nerves that lose these Schwann cell precursors with your manipulations. I wonder whether they change functionally in some way. Are they just a scaffold for these cells, or do they change functionally when these cells are gone, and then you also lose other signals that modulate in some way what they do when they reach the organs?
Yes, absolutely. Great question. There is a clear-cut answer to the first part of your question. So what happens to the nerves? These experiments they were done by the group of Jean-Francois Brunet at CNRS in Paris. And basically, they ablated genetically the entire neurocrest. So there is no neurocrest in the embryos. Of course, the innervation from the spinal cord still develops because ventral nerves that would innervate muscles and inner organs, they keep developing even in the absence of the glial component. Also, the nerves are developing that are cranial nerves. All cranial nerves are developing and branching. So for their navigation and for the principal development, nerves do not require neurocrest or they do not require Schwann cell percussors. Nerves are very autonomous from Schwann cell percussors or neurocrest. However, this is only valid until middle stages of development. Starting from embryonic day 15 approximately, all neurons that project to the periphery, if they don't get the assistance from the glial component at the periphery, they start to die. So they never make it entirely without the glial cells. They need glial cells for their survival. They can be generated, they can go to their targets, but then they will stay there for a little while and then they die without glial cells. This is the first kind of part of my answer. Second part of the answer goes to your question about whether the Schwann cell percussors can be different depending on the type of a nerve. So let's say if we are talking, can Schwann cell percussors inhabiting sensory nerve can be different from the Schwann cell percussors or Schwann cells, for example, inhabiting the motor nerve or like nerve depending on the position in the body. And this is exactly the project that we are finishing right now with single cell transcriptomics and different lineage tracing studies. And the answer is yes, they are different. There is a positional aspect. Mostly all Schwann cell percussors that associate with the nerves targeting inner organs, they are completely different. Well, not completely, but significantly different from the nerves innervating skeletal muscles and skin. And we think this is not because of the type of the nerve, but this is because of the local signals that dominate in the body during the development. So the inner organs, they prime Schwann cell percussors into the capacity to generate or regenerate autonomic nervous system. And that already sort of fate restricts, biases the Schwann cell percussors into that direction. So they have a chance only to differentiate into autonomic nervous system or to become myelinating or non-myelinating adult cells, which is very interesting. So it's not that it's only like sensory versus motor, but it's more kind of where in the body they are.
Thank you very, very much. And do you think that the neurotransmitters that those nerves release play a role in the process of differentiation or addition of these new cells to the target?
We think so. And we think that this is specifically important for the minor subtypes of Schwann cell percussors that will generate, for example, terminal glial cells at the neuromuscular junctions, and that will generate the sensory skin and organs like Ruffini and Pacini corpuscles, Meissner corpuscles, and so on, because they contain extraordinarily specific glial cell subtypes that we think are sort of primed or biased by the type of the neurotransmitter. And I think the beginning of the neurotransmission is something very important. Even for the sensory part, where the neurotransmission is not supposed to happen at the primary sensory location. Now we know that even sensory nerves, they can kind of release some minor amounts of neurotransmitters like all locations. And this sort of serves neuromodulatory and immunomodulatory and some kind of morphogenetic development or regenerative functions. Yeah, so I guess that's all I know about that.
Thank you very much. I really enjoyed the talk.
Thank you. If you have more questions, don't be shy. Okay. Then I guess this is it.
Igor, I have a question. So when I was in Sweden last time in 2012, we found some cells in the lungs. They are neurons that are close to the bronchi. So they should be also a Schwann cell precursor derived, right? But perhaps you can look at them.
Yes, we are sure they are Schwann cell precursor derived because there are, I think there is only one kind of large generalized neuronal type that is proximal to the glands, for example. These are parasympathetic neurons. And these all parasympathetic neurons in the body, they are Schwann cell precursor derived. For example, very important portion of Schwann cell precursor derived parasympathetic neurons are residing in the pancreas. So, and they are somehow important for the beta cells and the insulin release and so on. But we are not following this direction much these days, simply because we already published the concept that all parasympathetic Schwann cell precursor derived. So we don't think there will be anything like striking that we're going to find in that domain, except maybe for some molecular control of the process. And this is exactly where we are very weak, because studying molecular control is very hard, because I think this is the biggest problem of modern biology, because most of the processes in the body and during the development, they are encoded in the combinatorial way. It's not one molecule that does something, it's a combination of molecules. In order to understand the signaling and the regulation, one needs to screen so many things functionally at the same time in vivo that it just takes.
Yet, so these actually these neurons are not parasympathetic. They are like kind of sensory neurons that are just below the epithelium.
Below the
yes.
Below the epithelium. Well, I mean, like this will be something very novel. We also saw some cells with somas resembling the neurons, but I guess we can just discuss it, because this would be something very new. We are sequencing now skin, human and mouse, in single cell mode, and we also see some neuronal cells, although in the beginning we thought this is a contamination from the axons associated with Schwann cells, but we think there are primary sensory neuronal cells possibly existing in the skin, but we did not make a defined breakthrough in that direction, unfortunately. Maybe in the future. We will be very happy to collaborate with you, Jorge, depending on how you want to run it. It's a very interesting topic for us.
Okay, so no more questions. We are 71. Yes, go ahead.
I'm sorry, sorry, Jorge, to interrupt you. Thank you very much for this talk. It was really very interesting. I have two questions, which probably will sound very silly, but because I missed the exact beginning of the talk, or I didn't pay enough attention, I don't know if you mentioned something about it. But of course, this talk clearly focused on the development stages of any of these organisms that you have described. How much do you think that is retained in adult animals, or if it can be activated to induce regeneration of tissues or neurons that are lost in adult life? The second question related to this is, you focus and you observed all these kind of going outwards from the central nervous system to the periphery. Do you think that the similar mechanism can happen the other way around? Give a very simple example: DRG neurons that die after an injury of a peripheral nerve that could be reconstituted or regenerated backwards, so to speak, from these progenitor cells.
Yes. Yes, thanks a lot. Fantastic questions, actually, straight to the point. When it comes to the second part, I can tell we do not really know. I was working with people who run this dorsal root avulsion model, when people have this specific trauma, when the dorsal root is pulled out and many sensory neurons die. And basically the natural regeneration doesn't happen. It seems because of a lot of inflammation and a lot of connective tissue there. Maybe the principal capacity is there. So potentially cells can make it. But because it's like spinal cord regeneration, right? The axons love to regrow, everything can happen, but the scarring and inflammation, they sort of block that. And people have different level of success by blocking scarring and inflammation. And once they succeed better than others, neurons make the job, and the mice and rats, they're working and improving greatly, right? So again, I believe that the principal capacity might be there, but there are many other factors that contribute to the problem and kind of block this capacity from the full realization. But this is sort of like a long answer that can be simplified as I don't know. The first part of the question, I think, is also very interesting and important. We are trying to look into that. And we know a little bit more about the first part. So you asked basically if Schwann cell precursors can persist, let's say into the adulthood or late embryonic stages or postnatal stages, and they still can be plastic and they can contribute cells to different tissues and organs. We know at least one example where it happens and where it's not just happening a little bit, but it's very significant and very important. This is the development of the gut. So when the enteric nervous system develops, there are several sources of cells that contribute to the enteric nervous system of the gut. During very early embryonic development time points, the neurocrest cells, that is called vagal neurocrest, they will migrate massively and they will invade the gut and they will start to spread inside and build the enteric nervous system. However, there will be a second wave exactly in the same location. And this second wave will be huge amount of Schwann cell precursors that will multiply and propagate on the fibers of the vagal nerve, of the vagus nerve. So very early, once the first wave just whoop migrated into the gut, the vagus nerve becomes very strong, innervates the gut. And then there will be second wave coming with the vagus. And these cells, they will be recruited from the vagus, and they will give rise mostly to the enteric nervous system that inhabits our esophagus and largely stomach. So there will be sort of anterior part of the enteric nervous system. At the same time, in the back part of our body, the sacral stream of the neurocrest cells will migrate and invade the gut from behind, and then the streams sort of meet. So that's how the whole thing is completed. But this is happening still very early when the embryo is tiny and the gut is very small. It keeps growing with incredible pace. It's looping out, goes outside of the body. You've probably seen how the gut develops if you dissect the embryos of the mouse. And it becomes very big. And when the babies are born, they have extraordinarily fast pace of growth, and the gut is growing also with incredible speed. Imagine the gut of an adult human and the gut of a baby. That is newborn, right? It's a very big difference in size. And what is happening there, the extrinsic nerves, the visceral sensory nerves that come to innervate the gut during the growth, they contribute Schwann cell precursors, so more enteric neurons are seeded to accommodate for the gut growth. And in cases of aganglionic guts in Hirschsprung disease, what people see with the lineage tracing in all these mouse models, that yes, the gut is largely aganglionic, but next to these external fibers, visceral sensory fibers, specifically during postnatal time points, they start to receive the enteric nervous system in this aganglionic gut, but they never really manage it to do it efficient enough to achieve functional recovery. And all these extrinsic nerves, they of course bring these Schwann cell precursors, and most of these processes are happening during first 10 postnatal days. So it can even stretch into the postnatal times. We are not sure this is happening outside of the developing enteric nervous system, but for the case of melanocytes, for the case of pigmentation, for example, maybe some parasympathetic neurons in very specific minor places. Probably this is also happening and getting like around birth. And we are very much curious about the existence of the actual Schwann cell precursors similar to the neurocrest in absolutely adult nerve, just whether they are there in principle. So we keep sequencing adult nerves, trying to find the cells. But right now we don't see any. So we rather believe that Schwann cell precursors do not exist in adult nerves, but adult Schwann cells can revert back to the Schwann cell precursor states under specific conditions, because even adult Schwann cells, they are extraordinarily plastic because they can form reparative glial cells mediating the nerve regeneration and so on. And there are lots of gene expression programs and the modules that are shared between these reparative glial cells and Schwann cell precursors or immature Schwann cells. So still quite amazing.
But Igor, what happened with the odontoblast? What happened with the odontoblast? They are in the adults.
Yes, I completely yes, omitted that part because I knew I will not have enough time to talk about that. So we found something peculiar that during mouse tooth development and growth, because they have continuously growing mouse incisors, the Schwann cell precursors recruited from the innervation from the neurovascular bundle that is huge in the tooth. These cells can generate odontoblasts, the dentin building cells. The contribution is quite significant, but not major, so we don't understand the functional meaning of that, but it can be just sort of like residual evolutionary sort of capacity, because the cells stay very plastic, and given the conditions, they can separate from the nerve and convert into anything locally. Like we see it also for them, for the cartilage cells, for the mesenchymal component of the face. So we think it's sort of like a general phenotype that when the innervation is very abundant and the signaling is present, some Schwann cell precursors, they will just be recruited and they will just behave like normal neurocrest cells, but they will quickly differentiate into something locally. So because we do not understand the functional meaning of this, and the contribution is at the level that doesn't clarify the functional meaning, I decided that I will sacrifice this sort of topic, but the phenomena is there. It's quite interesting. Yeah. So these nerves and Schwann cell precursors, they do a lot of cool things, and even adult Schwann cells, they are very plastic and very interesting.
Okay.
Thanks a lot, everybody, for listening for so long to this.
An excellent talk. Igor, thank you for your excellent talk. It was very interesting and very innovative for us. We work in peripheral nervous system, but from now we have to change the name of Schwann cell precursor. They have to be another type of multipotent cell, because it would be difficult to explain to other people that you can do everything with the Schwann cell precursor.
Yeah, actually, thanks a lot for this very nice comment. We tried to change the name a couple of times, and every time we were just killed on the spot because, well, there was a very kind of powerful argument that still this is a very specific field of this multipotency in this Schwann cell precursors. Yes, it's all very interesting, relevant, and so on. But if we start changing this name, it's such a historical name, people from collateral fields or other fields, they will stop understanding which cells are we meaning, what are we talking about. It's sort of like, it's about being conservative in terminology. Sometimes we also want to stay conservative to make sure that people understand, everybody understands what we are talking about, right? So probably you noticed that the title of my talk was this enigmatic nerve-associated multipotent neurocrest-like cells, and I was like, what are these cells? Okay, probably Igor will tell us what are these cells, right? If I would say these are Schwann cell precursors, it would be way more clear. So after a lot of arguments with different reviewers, we decided we just leave it that way, and it will be enough if people are aware that Schwann cell precursors, they keep some degree of the multipotency of the neurocrest, and they are very interesting, plastic, very ancient population. Actually, I would like to spend the last 30 seconds saying one thing. We are also interested in evolution of cell types. We are going to publish a book on the evolution of the neurocrest. The book is called Evolving Neurocrest Cells. It will be out in July, covering all these topics. And it's not that I want to advertise the book, but what I want to say is that we think about the neurocrest, that neurocrest cells, they're sort of ancient cells that come out from the neurocrest, from the dorsal neural tube, and they do all these crazy things. They can migrate, they can differentiate into different options. But think about it differently. Think that on this migration and all these patterns of differentiation, you need so many complex programs. What if actually the primary way of dissemination was actually the migration along the nerves? Maybe Schwann cell precursors, they are the most ancient type of the neurocrest. And the free migration of the neurocrest is sort of already the sort of evolutionary later addition, is sort of evolutionary innovation that came after. And if we start looking at more kind of like ancient model systems like hogfish and lamprey, and I discuss a lot of this stuff with Marian Bronner and Shigeru Kuratani, who work with these model systems, we are coming to the conclusion actually that the primary mode of migration could indeed have been the migration along the nerves. That the neurocrest cells, they were exiting along the nerves, projecting from the developing neuron and reaching all the destinations where they would be locally recruited. For this, they would not need any navigation programs. They would need nothing except the signals converting them and differentiating them into something local. So, and now I think the field of evolution of the neurocrest cells is shifting towards the opinion that Schwann cell precursors, they represent more archetypical neurocrest, which is also quite unexpected, I think.
And we will be proud of our Schwann cell precursors to be so plastic and to able to do so many things.
Yeah, thank you. Me too.
Thank you, Igor. So
Yeah,
we hope
thanks
that you also come here to Argentina at any time to a meeting, maybe after the pandemic.
Yes, absolutely. I will be happy to come and see all of you in person and tell more interesting stories about Schwann cell precursors and everything else that we study in the lab. Okay. I hope all the crises will be over soon and yeah, it will be all fine. Okay. Okay. Thanks a lot. Then I will be leaving and have a good day, everyone. Bye.
Bye, thank you.
Unmute and clap.
Bye, thank you.
Unmute your mics and clap.
Okay.
Thank you.
Thank you.
Thank you, Jorge. Thank you very much, Lore, for the organization of the seminar. See you. Bye-bye.
Bye.